Hello everyone. Let's begin with today's session. On today in this course, Enzyme Science and Technology, we'll be studying about enzyme. We'll study about inhibition kinetics, different inhibitors, and how the kinetics of these inhibitors uh, occur. So, in the last class, we studied about inhibitors design, like how can we design different types of inhibitors, and today we'll learn about inhibitors kinetics. So, why do we have to learn about inhibitors kinetics? Because in order to understand how the inhibitors work, we need to understand the kinetics, how the rate of enzyme will, how the inhibitors will affect the rate of enzyme, and how it is affecting. So this will give us information about the enzyme mechanism and how the inhibitors can be used in order to study different enzyme mechanisms, and what are the roles of these inhibitors, what are the application of these inhibitors in inhibiting different enzymes. So we'll study about that also. So to begin, inhibitors are we already studied in previous classes that inhibitors are nothing but they are molecules that reduce the enzyme catalyzed reaction rates. So there are different classes of enzymes, different classes of inhibitors. These are divided essentially based upon two classes, whether they bind to enzyme either irreversibly or reversibly. So inhibitors can either bind reversibly or they can bind irreversibly. So let's take an example. So if we have enzyme and inhibitors, the binding could be irreversible. The enzyme inhibitor complex binding, it could be irreversible binding, or where it is not modifying the enzyme. And so in the irreversible binding, as you can see, you have both forward and backward direction. But in irreversible binding, enzyme is either modified by inhibitor or it forms a complex with enzyme inhibitor. With the enzyme, it forms a complex like this, and it is irreversible. That means once you add these irreversible inhibitors to the enzyme, they will bind to the enzyme irreversibly and make a complex, that is EI. They will make this EI complex. So so these are the two different inhibitors type, and there are different methods in order to study different inhibitors type. So what are different methods? So the first method, which we can differentiate either the inhibitors are reversible or it is irreversible. So the first method is a very common method, that is dialysis method. So in the dialysis method, if enzyme reversibly bounds to the inhibitor, then what will happen? That is, the inhibitor can exist in two forms. It can exist in the bound form or it can exist in the free form. So we can dialyze this free and bound form, and since inhibitors are very small molecule, okay, so they could be dialyzed out of this dialysis bag. So this dialysis bag will have different buffer concentration. The concentration of inside the bag is different as compared to outside. So this dialysis bag will elute out inhibitor, which is unbound to the enzyme. So from here we can check the activity of free enzyme as well, and we can check the activity from here, and we can find out that if we find any activity, that means there is a free enzyme available, and since there is a free enzyme, that means that inhibitor is binding reversibly. um, if it is irreversible binding, then all the enzyme molecules will be occupied by the inhibitor and there won't be any activity. So if we find any activity, that means that the enzyme and inhibitor complex is reversibly bound to each other. Similarly, we can do a gel filtration chromatography also. So in the gel filtration chromatography as well, we'll have a enzyme inhibitor complex, and we'll have a inhibitor, and we'll have a only enzyme. So these different forms of enzyme and inhibitors will give us different peaks. They will give us different elution peaks, and from here we can also confirm whether the inhibitor is completely reversibly bound to this enzyme or not. And the other method is dilution method. So this is not that sensitive method to find out the whether the enzyme is reversible or irreversible. That is, in this method what we do, we have so we particularly use some concentration, X concentration of enzyme and some Y concentration of inhibitors, and then we dilute this concentration as per our requirement. Like we can do a serial dilution where we can do it 1:10, 1:100, and 1:1000, and so on. And from this dilution we can check the activity at different dilutions, and this will tell us whether there is the binding is reversible or irreversible. So in the irreversible binding of inhibitors, as you can see here, these are the different example of irreversible inhibitors. These inhibitors, for example DFP, diisopropyl fluorophosphate, they irreversibly bind to the enzyme and they destroy the functional group. So let's take the example here. So this is difluorophosphate, isopropyl difluorophosphate. So we have two isopropyl groups and a fluorophosphate, and this fluorine here, so on the enzymes which has a serine in the active site, so the serine is the amino acid with OH group, and this OH will have a lone pair of electron, and that lone pair of electron will be taken by the fluorine in the diisopropyl fluorophosphate, that is DFP, and it will irreversibly get bound to this serine amino acid of the enzyme, and this serine amino acid is present in the active site. So that means it is modifying the amino acid in the active site, and this is a covalent modification which is actually destroying the functional group. So the OH is the functional group here, and that OH, then that the functional group is completely gone because of the DFP. So so this DFP inhibitor, it inhibits different enzymes which has this functional group. So one of the enzyme is acetylcholinesterase, and they have various enzymes which are commonly known as serine threonine proteases, like that of trypsin and chymotrypsin. So these enzymes have serine as their functional group, and this DFP molecule will modify this serine, and it will irreversibly inhibit these enzymes. So there are other irreversible inhibitors which are mostly heavy metals. So they also modify different enzymes. And then we have cyanide, which targets the cytochrome oxidase, so that's why cyanide is lethal to humans. And then we have fluoride. Fluoride also inhibits enolase, so it removes magnesium and manganese from the active site of the enzyme. So it forms the complex with magnesium and manganese, and it will remove those metal ions from the complex, and it will inhibit this enolase enzyme. So this is an example of DFP inhibiting acetylcholine esterase. So as you can see, we have acetylcholine receptors, and this purple molecules are the acetylcholine molecules. So what is the function of acetylcholine esterase? It converts acetylcholine to acetate and choline. Okay, it will cleave acetylcholine to acetate and choline. And this acetylcholine is responsible, it's a neurotransmitter, that means that it is present on the muscle receptor, and it is required for the contraction and relaxation of muscles. So if this enzyme is inhibited, then we could have problem in contracting muscles or relaxing muscles, which could lead to paralysis. So again we have the same inhibition mechanism here. So we have a hydroxyl group of serine on present in acetylcholine esterase, and this DFP, it will modify this serine molecule, okay, and it will lead to inactivation of this acetylcholine esterase enzyme. So this malathion is an insecticide. It is also responsible to inhibit acetylcholine esterase, so and this inhibition can lead to paralysis. There are other neurotransmitters which are inhibited by different irreversible inhibitors, like donepezil. So this inhibits the breakdown of acetylcholine again. Then we have other different inhibitors like RVT101. So these are the different neurotransmitters inhibitors which function for the inhibition further. So acetylcholine has different function apart from muscle contraction and relaxation. So so these are the molecules which are used as drugs and for the pharmacological inhibition of acetylcholine. So iodoacetate, as we studied in the previous class also, so iodoacetate will modify the cysteine group, that is the SH group on the cysteine amino acid. So this is also a type of irreversible inhibitor, or we can say chemical modification, where the SH group of the cysteine is chemically modified, that is the SH functional group is destroyed by iodoacetamide, and it leads to inactivation of the enzyme, as you can see here. So this is also an example, iodoacetamide is also an example of a irreversible inhibitor. So suicide inhibitors belong to irreversible inhibitors. So they have slightly different mechanism of inhibition as compared to regular irreversible inhibitors. So they are specialized form. They belong to the irreversible inhibitors class. They are also known as mechanism based inactivation. So why do they call this as mechanism based inactivation? So we have an enzyme, and then we have an inhibitor. So this enzyme and inhibitor, they will interact. So these are irreversible inhibitors, so we will not draw both forward and backward arrow, only forward arrow. So they will interact with each other, okay, and they will form a complex. And while this interaction, the enzyme will modify this inhibitor, Okay, the enzyme will modify this inhibitor. There will be some modification on the inhibitor by enzyme. So this enzyme will modify the inhibitor, and this modified inhibitor now will inhibit the enzyme, the free enzyme. So this modified inhibitor will inhibit the enzyme. So that's why they are called mechanism based inactivation. That means that in the same mechanism as enzyme is interacting with the substrate, enzyme interacts with the inhibitor, and it is modifying the inhibitor, and this modification leads to a special function, and that special function is it will inhibit the enzyme. Okay, so basically enzyme itself is modifying the inhibitor, and then inhibitor is killing the enzyme, so that's why the name suicide inhibitors. So these are the different properties of suicide inhibitors. They are irreversible inhibitors, and they are more effective. Um, they are synthesized with help of enzymes. So we can synthesize de novo suicide inhibitors based upon the mechanism of enzyme inhibitor or enzyme substrate interaction. Inhibition of xanthine oxidase by allopurinol. So we will see this example in the next slide. And this allopurinol is inhibited by so this allopurinol. So this inhibition of xanthine oxidase by allopurinol, and this allopurinol is used for the treatment of gout. So alloxanthine is synthesized by xanthine oxidase using allopurinol, is more potent inhibitor of enzyme than allopurinol. So we have, and there are different examples like 5-fluorouracil, it is an anti-cancer drug, and aspirin, which is an anti-inflammatory. So all these inhibitors are suicide inhibitors: allopurinol, aspirin, and 5-fluorouracils. So allopurinol, so as you can see, allopurinol is a part of purine synthesis pathway. And the byproduct of this pathway is uric acid. Okay, and this uric acid gets deposited in the bones. Excess of uric acid. So excess of uric acid can be get deposited in the bones, which leads to gout. So gout is a disease which is caused by uric acid deposition in the bones. So this allopurinol, it will modify this xanthine oxidase. So that this xanthine oxidase is the major enzyme involved in the purine biosynthesis pathway. Okay, and this it will convert allopurinol to oxypurinol. Okay, so and this will lead to so this oxypurinol, now this oxypurinol has the ability to inactivate xanthine oxidase. So that means this is our enzyme. Okay, this is our inhibitor, and this enzyme is modifying our inhibitor at here. Okay, it is modifying the inhibitor, and now this inhibitor oxypurinol, now it will go and inhibit the xanthine oxidase. It will go and inhibit the enzyme. So this is how the suicide inhibitors work. Then we have fluorouracil. So fluorouracil is also a suicide inhibitor. Fluorodeoxyuridylate, the full form, it's a suicide inhibitor, and it inhibits thymidine synthase. So this thymidine synthase is involved in purine and pyrimidine biosynthesis pathway, and in cancer cells which divide at higher which divide much faster as compared to the normal cells. So this fluorouracil basically will act on those um, rapidly dividing cancer cells by inhibiting the DNA synthesis of those cancer cells. So it's an anti-cancer drug. Now let's move on to reversible inhibitors. So in reversible inhibitors we have three types of reversible inhibitors. So we have competitive inhibitors. Okay, we have competitive inhibitors. Then we have mixed inhibition, and then we have uncompetitive inhibitor. So let's first understand competitive inhibition. So what is this competitive inhibition? So here, as you can see, we have so this is our enzyme substrate interaction, and which leads to formation of enzyme substrate complex, and then this will give you enzyme and products. But in case we add inhibitors, for example, this is our competitive inhibitor, it will go and bind to the enzyme. Okay, so this is our enzyme. So as you can see, the substrate this is the active site, right? This is the active site. So substrate and going, and it is binding to the active site, and enzyme is also binding to the active site, as you can see here. Inhibitor is also binding to the active site. So the geometry of the active site is complementary to both substrate and inhibitor. That's why in competitive inhibition, inhibitors bind mostly to the active site. Okay, they bind to the active site because of the geometrical complementarity. So the substrate will also so there will be a competition between substrate and inhibitor, that which one will go and bind to the active site. So this is the example of competitive inhibitor. So we have so this is the example where we have enzyme which converts succinate to fumarate, and as you can see, this enzyme has a geometric complementarity to the succinate. Okay, so the succinate is nicely fitting into this geometry, and it is converting succinate to fumarate, and the enzyme is released. So this is the general reaction. Okay, now the competitive inhibitor, which is a malanoid, and as you can see, it is resembling the succinate molecule, the substrate succinate, and it is also complementary to the enzyme, and it just does not have one CH2, okay, as compared to that of succinate. Okay, so they have a resemblance. So this malanoid will go and bind to this active site, okay, and this malanoid enzyme, it will not allow, basically it will not allow, it will inhibit the enzyme succinate, this formation of enzyme succinate complex, and thus it will lead to a competitive inhibition. So let's understand the competitive inhibition kinetics. So we have normal enzyme substrate kinetics, where enzyme is getting bound to substrate and it is converted to enzyme plus product. In case of competitive inhibitor, the inhibitor will bind to the enzyme, okay, reversibly. It will form a EI complex. Okay, so the total enzyme here is free form of enzyme plus bound to substrate plus bound to inhibitor. Okay, so this is the total enzyme which is distributed across different species, whether it could be free, bound to substrate, or bound to inhibitor. Okay, and we already know that the velocity for the rate of reaction, this is K1, K2. Velocity is directly proportional to the ES formation. Okay, and V max is equal to K2 ET. So when we'll get maximum velocity, when all the substrate molecules are in form of ES, that is, it will be ET. Okay, so this is our equation 2, and if we take a ratio of V versus initial velocity versus ET, what we'll get, we'll write this K2 ES upon what is ET. ET is this enzyme plus enzyme substrate plus enzyme inhibitors. So if we bring this here in the denominator, the K2, so the K2 ET as from this equation 2 is V max, so we can directly write V max, V0 upon V max, initial velocity upon V max is equal to, and let's substitute ES also. We can substitute ES, so it is S by Km upon 1 plus S. ES is S by Km plus I is I by Ki, because Ki is the rate constant of inhibitor. Okay, and if we solve this, we'll get V0 upon V max is equal to delta S Km 1 plus Ki plus S, and this fraction we can label this as alpha, so we can write this V by V max is equal to delta S Km alpha plus S. So this is the equation for the competitive inhibition. So this is the rate. So as you can see, the Km is getting affected here because it is multiplied by this alpha, which is actually the rate constant, which is the form of a rate constant of the inhibitor. So the inhibitor is affecting the Km here. Okay, so inhibitor is affecting the Km, that is the Km is getting multiplied by factor alpha, which means that, let's see how the graph will look like. So let's draw the graph. Okay, so this is the graph for without any inhibition, no inhibition graph of velocity versus substrate concentration. Okay, and this will be our Km, right, at half of V max. If this is our V max, V0 half of V0, the Km is equal to substrate concentration. Okay, and with inhibitor, how the graph will look like, it will look like this. Okay, the graph will look like this, and will actually, wait, let me try it again. This is how it will look like with inhibitor. So this is no inhibition, this is with inhibition. So if this is the Vmax, this will be our Km, and for this will be our Km. This is Km for inhibitor, and this is the Km. So Km for inhibitor is greater than Km. Okay, so the Km is increased in competitive inhibition. So let's see how the Lineweaver-Burke plot look like. So in the Lineweaver-Burke plot also we have reciprocal of Michaelis-Menten. Okay, so the slope is alpha Km by Vmax. Okay, and this is increasing concentration of inhibitor. Okay, and with increasing concentration of inhibitor, as you can see, the Km is increasing. Okay, Km here is in the denominator, so the Km is increasing. Okay, so these are the different drugs which are competitive inhibitors, and they are used for different purposes like cancer, in viral infection, and different diseases like hypertension. Okay, so we have 6-mercaptopurine. All these are the derivatives of purines and pyrimidines, and they are used to inhibit DNA synthesis or different enzymes like involved in the synthesis of purines and pyrimidine, like 5-fluorouracils, which we studied. It inhibits thymidylate synthetase. Okay, so these all inhibit the DNA synthesis process, and thus rapidly dividing cancer cells, and hence they are used for the cancer treatment. Okay, and we have neostigmine. It's a acetylcholinesterase inhibitor, and it is used for the treatment of myasthenia gravis. So these are the different examples of competitive inhibitors. Now let's move on to mixed inhibition. So in mixed inhibition, the inhibitor, this is the representation here. So we have a active site. Okay, so this is our active site, right? And the substrate is going and it is binding to the active site. Okay, but the inhibitor, it is binding here. Okay, it is binding here, which is not the active site. So it is binding to the allosteric site, we can say. It is binding to the allosteric site, that is any site other than active site. Any site other than active site. So it is binding to the allosteric site. So that means it can either bound to the free enzyme or it can bound to the enzyme substrate complex, because that site will remain free. So it will either bound to the free enzyme. So this is the free enzyme and this is the bound. Okay, so it can bound to both because this site is free for the binding. So it can bind to the free as well as the bound. So it is independent of the substrate binding. So this is a mixed inhibition, that is or non-competitive inhibition, that is it can bind, the enzyme binds to the site other than the active site. So it bounds to both free as well as the bound form of the enzyme. So as you can see, we have different rate constant for binding for either enzyme and enzyme substrate complex, and so let's derive this equation. How does it look? So we have again normal enzyme substrate E plus P. And now in mixed inhibition, we have inhibitors bound to either enzyme or both enzyme substrate complex, and with different rate constants, so K and Ki. So it will give us EI, enzyme inhibitor complex, or it can give us ESI, enzyme substrate inhibitor complex. So now the total enzyme here, it will be equal to enzyme substrate plus ESI plus EI. Okay, so this is how the total enzyme is divided. Okay, it can either be bound to substrate and inhibitor, and can either be bound to the inhibitor. Okay, now the Ki is equal to E plus I upon EI, that is unbound upon bound, whereas Ki prime, so this is Ki and this is Ki prime, this is equal to free of enzyme, free of inhibitor bound plus inhibitor upon ESI bound to enzyme substrate inhibitor. Okay, now let's write our V upon ET. If we take so we'll have here K2ES upon, so free form of enzyme will also be here, enzyme plus enzyme substrate plus enzyme substrate inhibitor plus enzyme inhibitor. Okay, again we'll bring the K2 here and it will become V max, because V max is K2 ET, right? So this will become V0 upon V max equal to substrate concentration. So if we convert ES into Km, we'll get Km 1 plus inhibitor upon Ki plus substrate into 1 plus inhibitor upon Ki. Okay, and then finally if we simplify this, we'll get substrate concentration Km alpha, this is prime, where alpha is equal to 1 plus I upon Ki plus delta S alpha prime, where alpha prime is equal to 1 plus I upon Ki prime. Okay, so this is the kinetic equation of mixed inhibition. So how the graph will look like. So it will have the effect on V max also now, in case of, so this is V0 upon substrate. So now V max will also be affected here. The V max will decrease in V max. And this is the Km. For here the Km will be this, and the Km is decrease. Okay, the Km for inhibitor is less than the Km. Okay, and Vmax is decreased. So both Vmax and Km are decreased. And how the linear curve plot will look. So so this is the increasing concentration of inhibitor. Okay, this is no inhibitor, and if we increase the concentration of inhibitor, as you can see the Vmax here, Vmax is increasing, but it's in the denominator. Okay, so the value of Vmax will decrease. So the Vmax will decrease. Similarly, Km, okay, Km will also decrease. So this is the example of mixed inhibition. So we have different heavy metal inhibitors like Ag2 plus and Hg2 plus. Okay, these are inhibited by these inhibit the binding of SH in the enzyme. Okay, so these are all the examples of mixed or non-competitive inhibitor. So we have pepstatin, which inhibits the enzyme pepsin. So again we have a representation that this is the active site and the inhibitor will bind to the site other than the active site. Okay, and after binding to this allosteric site, there will be some conformational change which will not allow the substrate to bind to the inhibitor. Okay. So what is the difference between competitive and uncompetitive? First of all, in competitive the inhibitor will go and bind to the active site, whereas in the non-competitive it will bind to a allosteric or a regulatory site and it will modify the active site. Okay, so these are the examples of and representation that how competitive and non-competitive work. And next we have uncompetitive inhibitor. So in case of uncompetitive, as you can see, the inhibitor is binding to the enzyme substrate complex. So whenever substrate binds to the enzyme, there will be some modification in the regulatory site which will leads to the binding of the inhibitor here. Okay, so let's derive the kinetics. We have enzyme plus substrate giving us enzyme substrate, and the inhibitor binds to the ES complex giving us ES high. Again now the total enzyme here would be free enzyme plus bound to substrate plus it could be bound to inhibitor also, and Ki, so we'll have only one rate constant Ki. Okay, Ki for ESI will be enzyme substrate concentration upon inhibitor into inhibitor upon ESI. Okay, then again we know that K2ES, but if it is Vmax it will be K2ET. All the enzymes will get converted to ES, so we'll have a maximum velocity. So if we take V by ET, if we take the ratio of this, we'll have K2ES upon ET is this E plus ES plus ESI. If we bring K2 here, we'll have V0 upon Vmax again, and we can substitute ES as substrate plus upon Km upon enzyme simplify this, we'll have S by Km 1 plus S by Km plus S plus I by Ki, and this will give us a final equation. Km alpha, so we are considering this as alpha. So this is the kinetics for competitive inhibitor, and this is how the Lineweaver-Burke plot will look like for the competitive inhibitor. So as the so both Km, so here Km is decreasing and Vmax, as you can see, Vmax is also decreasing. Okay. So this was this is what was taught in the enzyme inhibitor kinetics, and now we move on to the assignment questions. So the first assignment question is, if the substrate concentration is much higher than the Km, Michaelis constant, what is the relationship between reaction rate V and maximum velocity? So first let's write a Michaelis-Menten equation. So we have V0, okay, that is reaction rate, Vmax, substrate concentration upon Michaelis-Menten constant plus substrate concentration, and in the question it is saying that Km substrate concentration is much higher than Km, that is we can just ignore Km. So the Km here is very small, so we can just ignore and take it as, we can approximate it as 0. So what it will become, Vmax S upon 0 plus S. So this will cancel out, so V0 will equal to Vmax. So correct answer is V equal to Vmax. Okay, so next question. The maximum velocity Vmax of an enzyme catalyzed reaction is 50 micromole per minute, and Michaelis constant is 10 millimolar. Calculate initial rate when substrate concentration is 20 millimolar. So again we'll write V is equal to Vmax plus substrate concentration upon Km plus S. So we need to calculate V0. Okay, so this is 50 into 20 upon 10 plus 20. Right, this is our substrate, this is our Vmax, this is substrate, and Km is 10. Km is 10. Okay, so this will be 1000 upon 30, which is 33.33. So correct answer is 33.33 micromole per minute. The catalytic efficiency Kcat by Km of an enzyme is 1000. Molarity inverse, second inverse. If the kcat value is 500 second inverse, calculate km. So very simple. So kcat by km is the catalytic efficiency. They have already given us, it is 1000, and they have already given us kcat also. So 500 upon km into 1000. So km will be 500 upon 1000, which is 500 upon 1000, right? Km, we need to calculate km. Yeah, so it will be 500 upon 1000, so 0.5 millimolar. The turnover number kcat is an enzyme of an enzyme is defined as maximum reaction rate, the ratio of Vmax to Km, the number of substrate molecules converted to product per enzyme active site per unit time. Yes, so the correct answer is this. Correct answer is the number of substrate molecules converted to product per enzyme active site per unit time. The type of enzyme inhibition that can be overcome by increasing substrate concentration is competitive inhibition. So competitive inhibition, as we have studied, the inhibitor bind to the enzyme's active site, right? So okay, it will bind to the enzyme active site. Okay, and that active site will be competed by the substrate also. So there will be competition to bind to the active site. Okay, and this competition can be easily biased by adding more substrate. So if we add more substrate, there will be less inhibitor molecule attached, so there will be less inhibition. So we can overcome competitive inhibition by just increasing the substrate concentration. The purpose of using double reciprocal Lineweaver-Burke plot in enzyme kinetics is to directly calculate Vmax, directly calculate Km, to identify the type of inhibition, to determine the reaction order. So we can identify the type of inhibition based upon the graph what we look like. So if we use different increasing concentrations of inhibitors, there will be different kind of graphs. For example, in case of uncompetitive inhibitor, the graph will look like this. Okay, in case of uncompetitive, the graph will look like this. In case of competitive, the graph will look like this. Okay, so the graph pattern will change based upon what type of inhibition is. So we can identify the type of inhibition also from the double reciprocal Lineweaver-Burke plot. Okay, so this is the correct answer. So we have one more problem. Vmax is 40 micromole per minute, Km is 10 millimolar. Substrate concentration is 15 millimolar. Following Michaelis-Menten equation, calculate the initial velocity. So initial velocity V0 is Vmax, substrate concentration upon Km plus substrate concentration. So Vmax is 40 into substrate is 15 upon Km is 10 plus 15. So this will be this will be 600 upon 10 plus 15 plus 25, right? calculate this. So this will give us 24. Okay. So but it is not in the option here. But the correct answer is 24. In reversible non-competitive inhibition, inhibitors bear structures resemblance to substrate. Inhibitor lowers maximum velocity attainable with given amount of enzyme. Km is decreased, Km is increased. So in reversible non-competitive inhibition, the inhibitor bears the structure resemblance to no. It doesn't have to have structure resembling. Competitive enzymes to have structure resembling to the substrate. Inhibitor lower the maximum velocity attainable with the given amount of time. Yes. So it will the Km is decreased, the Km is increased. So in case of non-competitive, the inhibitors lowers the Vmax. Okay, so the correct answer is this, B. The enzyme reaction follows Michaelis-Menten kinetics. The reaction velocity at substrate concentration Km is Km by 3 is dash. So let's write Michaelis-Menten V0 into Vmax substrate. Now the substrate concentration is Km by 3. So they are saying S is equal to Km by 3. So let's substitute this value here. So V0 is equal to Vmax Km by 3 upon Km plus Km by 3. So what we'll have? We have Vmax Km by 3 upon, so this will be 4 Km by 3. Okay, so we can just cancel this Km by 3 and we'll have V0 is equal to Vmax by 4. So here this is our correct answer. In order to catalyze reaction, an enzyme is required to be saturated with the substrate, decrease the activation energy, increase the activation energy, increase the equilibrium constant. So the enzyme doesn't affect the equilibrium constant at all. It has to decrease the activation energy. So the correct answer is this. It has to decrease the activation energy. Zymogen in the active form of enzyme. Zymogen is the active form of enzyme, complex form between enzyme and substrate, inactive form of an enzyme activated by cleavage. Yes. So zymogen is the inactivated form of enzyme that is activated by the cleavage. So we have different zymogens of chymotrypsin and trypsin. So they are activated when they are cleaved. So those are the zymogen forms. In Lineweaver-Burke plot for an enzyme catalyzed reaction, the x-intercept is So the x-intercept is minus 2 millimolar and the y-intercept is so this is the equation, double reciprocal Km Vmax substrate concentration plus 1 by Vmax. Okay, so the x-intercept is minus 1 by Km. So if we take a mod of this, we'll have minus 1 by 2m. Okay, so which is equal to 0.5 millimolar. Right? So in the Lineweaver-Burke plot, the x-intercept is minus 2 millimolar. Minus 2. Okay. Oh, sorry. So this is minus 2. So 1 by Km is 2. So Km will be 5. So the correct answer is 5. Lower value of Michaelis-Menten constant shows greater affinity to the enzyme for substrate. Yes, it shows greater affinity. Lower the Km, higher the affinity. The greatest increase in the reaction rate will be of increasing the substrate concentration by factor of 1.5, by factor of 2, by factor Michaelis constant. So if you increase the maximum velocity by the factor of 2, that will increase the rate. So just increase the maximum velocity, the fourth option. The ratio of V0 by Vmax when substrate is 5 km is, so again same type of question. V0 by Vmax is equal to Km plus substrate. But substrate is 5 km. So this will be 5 by 6, right? Km. So 5 by 6. Okay. So this was the end of today's session. Thank you.